Hey everybody, we are going to finish up talking about DNA, and we are going to look at how DNA is packaged. And what I mean by that is how chromatin is packed together. So we're going to look at 4 different things. The focus is going to be on number 1 and 4. So first we're going to look at nucleosomes, then we're going to look at chromatin fibers, third is looped domains, and then 4 is chromosomes. So again, we'll focus on nucleosomes and chromosomes, but this all has to do with the way that DNA is organized. So here we have our DNA double helix. Okay, so this is just a really long molecule. And you can see what's happening is we have these molecules right here called histones. And what's going to happen is the DNA is going to loop around 4 histones, and it's called a nucleosome when that happens. And so when you have a strand of DNA that has started to loop around these histones, we say it looks like beads on a string. And so that's kind of what you're looking at here. So you have very visible nucleosomes. So again, that's going to be a strand of DNA wrapped around these proteins. And then you're going to have a 5th protein called H1 at the base of that. So nucleosomes, again, kind of look like beads on a string, and it's DNA that's wound around a protein core. Those proteins are the histones, and this is essentially a way to package DNA, and it controls how genes are read, how often they're read, how quickly they're read. The protein core are 2 molecules of 4 types of histone proteins, and then you have H1, which is a 5th type of histone protein that is going to attach to the DNA outside of that core. Next we have the 30 nanometer chromatin fibers. And this is a cylinder of tightly coiled nucleosomes, and it's going to be about 30 nanometers in diameter. So what am I talking about here? It's this guy right here. So this is going to be essentially a fiber of nucleosomes that are grouped together. And we skipped over looped domains. So those 30 nanometer fibers are then going to create these loops that you can see right here. And so those are the looped domains. And then the looped domains are going to pack together to make a chromosome. So chromosomes are large units of DNA. This says they're similar to chapters in the Book of Life. And then you're looking at what's called a karyotype down here, and this is actually an abnormal human karyotype. It's abnormal because it has 1 extra chromosome. So you have chromosome pairs 1 through 22, and then for the 23rd pair of chromosomes, normally if you're looking at a female, you would have 2 X's, or if you're looking at a male, you would have an X and a Y, and here you're actually looking at 1 X and 2 Y chromosomes. And so these chromosomes all contain really important DNA. This is something that's done very frequently, karyotyping, when you're looking for genetic disorders. And so if a chromosome is missing or part of a chromosome is missing, that is a pretty big deal. So think about if a chapter was ripped out of your AP Bio book, you would obviously be missing a lot of information. Same thing with these chromosomes. If you're missing a chromosome or a part of a chromosome, that has some pretty big implications. So the summary for the DNA chapter. You need to know the scientists and their experiments. This is something we worked a lot on in class. You need to know why DNA is an excellent genetic material. Again, that ties in very much with that first point. How DNA replicates. So you should be able to kind of walk through all the steps and stages. You should also be able to talk about all of the enzymes that are really important for DNA replication. You should be able to discuss problems in replication, so any errors and if the errors can be fixed, how they're fixed, what happens if they aren't fixed, and then finally chromatin packing, which we just talked about today. So your lecture question, why could it be beneficial for DNA to be organized into chromosomes during certain times but not others? So I want you guys to think about that and shoot me an answer via the website. Thanks guys.